Similar aspects for quite some time, but it got formalized. And for past few lectures, I have been covering this particular aspect. And when the Dr. Ravindran asked me that this would be the topic, I thought probably, I would be able to speak something about it. You all know that this particular aspect is important, but we are working and we have generated some data, but I will not be presenting very much of our data. Most of it is unpublished. I'll give you general outline of what we are doing and how we are doing. And more importantly, I will be talking about the approaches and briefly about challenges and opportunities that we have before us to prospect new genes and alleles from a wider resource that we have in this country. Okay, so prospecting is nothing new. Bioprospecting started in prehistoric time. The people did not know what they are prospecting to start with, but the focus was to identify something which would be useful as food, useful as medicine, and then in fact all the domesticated crops that we have today, they are actually prospected from the wild. So rice we have, we don't think about it, but they are all prospected by our forefathers, our ancestors, for the purpose of food, for the purpose of medicine. You see the wealth of medicinal plants that we have in this country. So they are all prospected. Which one would be useful, which one is not, and which one is useful for which purpose. So identifying those was a Herculean job. They must have really spent quite a bit of time and kept on trying, because the systematic analysis and all that was not there. So they must have kept trying actually. Thousands of different herbs for a particular ailment, and then finding out ultimately that this is the herb which is curing this particular disease. This is basically prospecting a particular species for a particular purpose, a particular organism for a particular purpose. Informal bioprospecting began in the prehistory people, as I said, and then we found out many useful plants and their uses we determined. Scientific bioprospecting started much later, and we did this to identify active ingredient. As I said earlier, it was total leaf, and today we say, okay, this is the active ingredient in this particular leaf, in this particular species, which is important for curing this particular disease. So not only the organism, but also we are identifying the biomolecules which are important for a variety of purposes. And this is one case of an accidental discovery. So prospecting a particular molecule, which has today, which at that time had far-reaching consequences. People were dying, and then with these antibiotics, and one could control infection. And subsequently, we have an array of such antibiotics which were discovered subsequently and put to use. So this is a beautiful example of accidental bioprospecting from a microbe. First of all, identifying the microbe, and then subsequently putting that to use. And beautiful example. And you have now, hundreds of such examples of bioprospecting. Our focus today of discussion would be prospecting genes. We all know genes are important, and we know that our DNA carries the genes. Ultimately, we know RNA has genes, but then for a large part, rather the whole part, will be concentrating on DNA carrier of genetic DNA. We know that unknowingly that we are prospecting genes. We have already prospected genes, even knowing what genes are. For instance, a beautiful example is dwarfing height of rice or wheat. Beautiful example of prospecting a wheat and putting that to use. We don't realize that we have prospected a gene, but basically we have prospected a gene in the form of dwarfing genes in rice and wheat. So, and then you know that what kind of revolution they have brought in terms of the revolution that we talk of. Just one gene in rice and one gene in wheat, or one or two genes in wheat. So that has brought great revolution in this country. So bioprospecting of genes aims at obviously discovering those genes from biological resources, diverse resources. And because we know from this example that you can bring a gene from bacteria and put to use in plants. So there is no sexual barrier question remaining anymore. So that you can go across sexual barriers and identify genes from any source and transfer to any target organism. So that's a beautiful example how another revolution can brought about. Although your government, I learned yesterday, that banned all work on transgenics, I believe. I do not know what extent. Farmers cannot be stopped. Actually, BT cotton came to this country because of farmers. It got commercialized because of farmers. So if they find something very useful and then they can save money, they can get more benefit, or there is more of health benefit to the consumers, I don't think anybody can stop. The governments cannot stop. The whims and fancies of people, cannot really stop something which is very, very promising and which can benefit a larger section of the society on the whole. So it's very difficult to prevent that, and that's what happened actually. BT cotton, a beautiful example of prospecting of a gene from a microbe and transferring that to across sexual barriers to plants, and many different plants. And here we have BT cotton, as an example for us, bright example for us, that a bioprospecting can help and to a great extent solve the problem, and a serious problem. At a one stroke, we have solved the problem of higher cost of production. We have solved the problem of pesticide use and then the other negative effects of pesticide use to environment, to human health, and all that. So at a single stroke, many problems could be solved, and it's ecologically more safe than the transgenic itself that many people question. That's a beautiful example of bioprospected gene. That's what I wanted to highlight. You have also some other genes you find mentioned in literature, prospected from the microbe like bacillus, like yeast, like E. coli and other organisms, and even plants like moth bean, the Arabidopsis, the wheat. So you have also other examples, not really commercially exploited to the extent it has been done in case of BT cotton, the BT genes, but they are being tested and retested rather in many different systems, and hopefully some of them would click. And similarly, there are many other genes which are being worked on worldwide, and they are being tested, and many diverse sources have been really utilized to isolate such kind of genes, prospect genes. There are many different approaches available. As scientists working in molecular biology or students of molecular biology, you are all aware of this. Because the topic says that we have to talk about it, I will take you through very briefly some of those approaches which we are employing currently to prospect genes from very diverse resources, and you will see what are resources we are using towards the end. We all started, molecular biology all started with the use of heterologous probes and primers to clone a gene, and also protein route to isolate genes. Either screening cDNA library, which has been made in expression vector using either antibodies as probes from purified proteins, or using oligonucleotides to screen cDNA libraries, or even genomic libraries. So they were the ones which were used in the initial stage of molecular biology to clone genes. In fact, the first examples of those cloning genes. If there is a protein which you can purify, which is expressing at a very high amount, like seed storage proteins in case of plants which were worked on to start with, so you can purify them and then partially sequence them and design some probes, degenerate oligos, and then screen the library. Alternatively, you can go for designing antibody probes and then screen the cDNA library in expression vector and then isolate the gene. They were the early examples of isolating genes where you do not know anything. If you know something in some system, you can use a heterologous information, either in the form of primer or probe. You can design and clone the genes. But if you do not have this information available, then currently the emphasis is on profiling the transcriptome of a particular organism, comparing under different situations, different tissues, different organisms, and different plants, and then identifying those which would be of use. Alternatively, one can go for employing a tool called insertional mutagenesis. So you can go for inserting a foreign element into a gene, inactivate the gene, mutate the phenotype, and then subsequently from phenotype and also from the sequence that you have inserted into the gene sequence, you can reach the gene sequence, you can clone the gene sequence. And worldwide there are many efforts, and it all started isolating genes. Although they were discovered in maize, the transposable elements, and they were put to use to a great extent in case of bacterial systems. Many genes were cloned using this transposable mutagenesis. And today you have tDNA inserts and mutagenesis, and then also retrotransposable mutagenesis to isolate genes from plants. And so you have many different examples. And if you see the leading journals like Nature, Science, Plant Cell, which are Living Cell, which are publishing gene isolation, you'll find these three approaches are utilized to a great extent. And every month papers are published in very high impact factor journals utilizing these approaches. And also, of course, you have a map-based approach, how you can use linked markers, and you will see briefly I'll explain this for the purpose of giving you some message that how these approaches work actually. And also integrated approaches, you can combine particularly the transcriptome profiling and the map-based cloning approach, and which greatly facilitates and reduces your effort to isolate genes and cross-link genes from resources which are very diverse in nature. If you think in today's context, this is the approach which is extensively being utilized to isolate genes. And practicals will be doing something like PCR, RT-PCR, real-time PCR, and all that, which are subsequent steps. Once you identify something using these strategies, then you go for very fine dose. And do they really express differentially? If they are expressing differentially, do they really have the required function? Then you have to validate that in transgenic system. So these are the I have just outlined. I have just listed these kind of methods which are currently in use to isolate genes, identify genes first of all which are showing differential expression. The differential display RT-PCR, real-time PCR, reverse transcriptase PCR, then cDNA FLP, representational difference analysis, suppression subtractive hybridization, and also you have microarrays, particularly DNA microarrays which are being extensively utilized. In fact, the world is coming to close on this. In fact, to close this particular aspect of work rather. So microarrays particularly. Not many labs are actually working currently on microarrays. But anyway, so they're still being utilized to some extent. Then serial analysis of gene expression again had a great promise, but slowly it's going off the shelf. Then massively parallel sequencing, signature sequencing, or simple sequencing. Massively parallel sequencing is coming in a big way to identify genes which are showing differential expression between organisms, tissues, and stage of development, and whatever you want to identify. And I'll briefly say, one particular approach how we are using in our particular program. This is one which we are extensively utilizing to identify genes which are showing differential expression. This is one which was prior to this. Also, there are some description of the technology, but very vividly described and put to use in this particular paper published in Proceedings of the National Academy of Science in the year 1996. This is suppression subtractive hybridization. Very simple principle, but here you don't have to really make cDNA libraries to go for subtractive cloning of genes, which you used to do prior to this particular. And more importantly, you can enrich the genes which are showing differential expression and clone those. So that's another advantage of this particular technology. And they're all written down below, so I'll not read each one of them. But basically, this is a method to identify genes which are differentially regulated, and obviously that is reflected in the differential expression of these genes. And the approach is given here, and I don't know, time would not permit me to really discuss each one of them. So basically what you do if you want to identify genes which are expressing in this particular taster sample, which is called taster cDNA that you synthesize here, and you use one particular adapter. And then you have also, that you make two parts. This is part 1, this is part 2, and ligated with two different adapters as indicated here. This is the black box here, this is shaded box here. So you have two different adapters, they are getting ligated to the 5' end of this. This is the 5' end, and you can have 5' end ligation, and this is ligated here, this is ligated here, and 3' ends are free. So this is the same sample. So you have taken, suppose you are isolating DNA from flower tissue and the leaf tissue. And if you want to really isolate genes which are expressed specifically in flower or at a higher level in flower and at low level or not expressing in leaf, so leaf is your driver, cDNA. So whatever RNA, messenger RNA you isolate, convert that to cDNA from leaf, so that becomes your driver. And then you isolate RNA, messenger RNA, convert that to cDNA from flower, that becomes your test. Now you have to remove all that which are common. So for that you need to hybridize. So you synthesize this double-stranded cDNAs and denature them, and then hybridize with the driver. So driver, test, or hybridization takes place. So that means all those which are common, so they will be hybridizing to each other. Okay, all those which are common, they will be hybridizing to each other, and that's what you see. So they will be hybridizing. So this, in tester you have no adapter ligation, so they are all tester driver hybridizations. Tester driver hybridizations. Some of them might remain single-stranded, and they are self-hybridization. So the same molecule coming back, Okay, and this is the driver which is not hybridizing, and this is the driver, and again coming back. This is the driver, and the separated molecules in the two strands, and these two strands are coming back together. So this is what you find, after hybridization of tester with driver, and you can do that for 2 times to ensure that all that which are common, they have gone into hybridization between the tester and driver. Nothing escapes. That you can ensure, and you can repeat that in thrice and see that most of those which are common, they're all made into this kind of hybrids. And after that, what you can do is you can denature and mix them. These two you can denature and hybridize to each other. So what will happen in the process is that this one and this one can hybridize. That's what you see here. That's the additional hybrid that comes out. So this one and this one can hybridize. They are already there and they are already there, so they will be all there. So when you denature and again renature and reannul, so you have another class which will be coming out of this, and this is the additional class which comes out. And then what you do is you go for clenophenene. If you use enzyme, cleno enzyme, and also add DNTPs, so the 3' end, this is a 3' end, and it will be extended because you have the corresponding complementary sequence, it will be extended. So you will have filling of these overhangs which are there, so they will be filled there, and then you will have a double-stranded molecule which is available now. And this is the double-stranded molecule which will be actually amplified, and that is the one which is going to be cloned. And that is the beauty of this particular technology that you can employ. That you don't have to really have cDNA library, but all those which are getting amplified, you can clone them. And most of them, there will be some escape still, most of them will be actually the genes which are expressing at a higher level or only in tester tissue, that is flower tissue, as compared to the driver tissue, that is the leaf tissue. And the others, particularly say in this particular case, this will not be amplified. As you know, this is 5' end, this is 3' end, because your primers will be corresponding to this particular sequence, that is your adapter sequence, this particular sequence. And you don't have 3' end here, so your primer would not bind and amplify this one. Because as you know, your synthesis will be only when the primer binds to this particular end and synthesizes. In this case, it will be linear synthesis because your primer will be binding here. This is 3' end, this will be binding here, and then synthesizing. So linear synthesis of molecule. And in this case, there is no primer binding site, so it will not be actually PCR amplified. And this is the one which will be amplified. And in this case, there will be primer binding this side and this side. This will be one primer binding. The other, this is not there. So there will be same adapter ligation. So what will have is a kind of structure which is called frying pan structure. You can see here, and this structure doesn't allow PCR to work, and this is called suppression PCR. The suppression PCR will eliminate those which are only present here and making this kind of molecule. So the ones which are hybridizing from here and here, so they are the ones which will be amplified. So indicating that they are the molecules which are only present in tester, not in the drive. So this is what you identify, and that's what they did. You can see in this particular paper, they identified a gene which is expressing only in testis, nowhere else, because they have taken tissues from many different organs, and they find that one particular gene expressing only in testis, nowhere else. And that is how you can identify tissue-specific genes, development stage-specific genes, and also abiotic stress-specific genes, induced genes. if you impose salt stress and then see what is happening under control condition, what is happening under stress, and then you can compare. If you take a resistant genotype and compare that to a susceptible genotype, you can have library both in resistant and susceptible, and then